Dr. Sangiorian, thank you so much. Very kind. And I first apologize to all of you for my English. My Italian is very poor, so I beg on your indulgence. Grazie. I also want to thank Dr. Ungaro, Dr. Gambrini, and Dr. Galimberti for the extraordinary presentations that you all have given, allowing me to talk a little bit about how epigenetics ties together with the chronic disease epidemic that we're experiencing in the world today. And it is truly that of a global epidemic. In 1990, I was invited to speak at the Hong Kong University Medical School. And I chose to speak on type 2 diabetes because I felt that it was an important rising chronic illness in the world. After I finished my presentation, the dean of the medical school came to me and he said, "Professor Bland, very nice lecture. However, we don't have type 2 diabetes in China." In 2009, a mere 4 years ago, I was invited back to the same medical school. And the request was to talk about the epidemic of type 2 diabetes in China. In literally less than 15 years, China has now become the country with the largest number of type 2 diabetics in the world. 214 million type 2 diabetics. You cannot explain that disease prevalence, the increase in disease prevalence, on the basis of Darwinian medicine. Let me say it again. You cannot explain the origin of that increase in type 2 diabetes in that mere 15 years on the basis of Darwinian medicine, meaning genes that mutate that then become more susceptible and you have something. You can only explain this on the basis of what is like an infectious disease, which means epigenetic. The genes have not changed. What has changed is the environment in which the Chinese people are living. It's the universal modifier of their function. I use the word function. It's the basis of how we redefined function in 1990 when I established the Institute for Functional Medicine. People have asked me, wasn't that a poor choice of words? Because function in medicine already has a definition. It's called like geriatric medicine, meaning disability in older age, or psychosomatic medicine. It's all in your mind. It's a functional disturbance. That was the connotation that function had when I decided in 1991 to name this institute the Institute for Functional Medicine. Why would I choose a name that was already predefined? Because when I was reading the literature, going to meetings, talking to leaders across many medical disciplines, and I started to see that radiology had functional MRI, that cardiology had functional cardiology, that endocrinology was talking about endocrinological function, not just endocrinological pathology. And I said to myself, maybe this is the forward-looking concept that's going to anneal different disciplines into a singular thought about how to beat back the rising tide of chronic illness. Because if you wait till you have a diagnosis, it's a very late stage to try to do something. And at that point, you're into heroic medicine, pharmacology, surgery, hospitalization, very costly, and with limited, really successful outcome. If we're real about medicine, and I was a medical school professor, researcher for 15 years, so I looked at myself in the complex of my colleagues, and I said, what are we really delivering at all this expense? With expensive hospitalization and tertiary care, We're giving a lot of people a sense that something's being done, but if you look at outcome, are we really delivering cost-effective outcome? And I came to the conclusion, no, there had to be a better model. The model that we were using was great for a crisis that related to a trauma or an emergency room event, but it was very poor in delivering successful, evidence-based, improved outcome for people with chronic illness. So that was my audacity to take the term function and redefine it into this new model, where genomics intersects with lifestyle. Now, what does that mean in terms of a healthcare model? All of you, I'm sure, have this kind of a concept in mind, or you teach your patients about it, or it's your thought process. And this is just another way of saying that disease normally doesn't occur out of nowhere. You don't get a heart attack on Monday morning when on Sunday your heart was in perfect health. Now, you may not have known you had some problem with your heart on Sunday, but Monday, when you had the heart attack, suddenly you were reminded something was not right. The concept which underlies everything I'm going to talk about from here on in the next 50 minutes is the questions you ask determine the answers you get. If you don't ask the question, you never get the answer. So if you never ask the question about does that person have dysfunction, then the answer is they must not have had dysfunction because you never asked the question. The heart attack just occurred. This model of disease care is related a lot in business to what I call the cost accounting vision or strategy in business, which is where you let accountants run your business. When an accountants run a business, what they do is they audit the business, and they look at how the business did the past year. It tells you nothing about where you're going. It tells you nothing about how successful you might be. It just says in the rear view mirror of life how you were. A forward-looking medicine is not an accounting kind of medicine. It's a entrepreneurial business building kind of medicine. It looks forward to where the prospective steps or journey or trajectory of that patient is taking them, or that person, before they arrive at that ultimate event where they have a clean diagnosis of a critical episode in their life. So that's what this particular model talks about. You could call these pre-pathological stages, or preclinical stages, or precedent stages, or disruptive stages, or dysfunctional stages. But it's prior to crossing that boundary to a clean diagnosis. That's the stage where less aggressive intervention can be successful. It's the stage where the patient can participate in their own program rather than being laid on a hospital bed. They can actually participate in the program. And I would pose from multiple studies, it's the one place where you can save vast amounts of money in healthcare by preventing that, what I call horizontal disease. Horizontal disease is where you're laying flat on your back and someone's asking, what are we going to do to save that person? Very expensive when you get to that point. So this is a different kind of healthcare model than the one that we have been practicing, applying, and paying for in the world for the last 40 years. There's nothing wrong with what we've been doing. And in fact, logically, what we've been doing is the right first step in the developing of a medical system. Because in a triage system, triage meaning handle the big problems first, the first you're going to do is develop a medicine to handle the most acute care problems. Then, as your disease patterns change from acute care to chronic care, your model needs to change. The problem is, the model hasn't changed. We're still trying to pound a square peg into a round hole. Italy, France, America, China, I don't care where you go in the world, the model that we're using presently to manage the rising global burden of chronic disease is inappropriate to be successful in improving outcome at a cost-efficient level. It will not work, I guarantee it. The pill for the ill, single diagnosis against single agent of treatment, is not a model that will ever lead to cost-effective outcome. Why? As you've heard from my previous experts in this presentation, because these diseases don't come from one origin. It's not like pneumonia caused by a pneumococcus bacterium for which one pill called an antibiotic will treat it. That's a very good model for that particular kind of disease. If you have 100 type 2 diabetic patients, you have 100 type 2 diabetic patients. No two of them are identical. So if you try to use a single model of a pill for every diabetic, you're going to be absolutely unsuccessful in getting positive outcome. And we see that. This is called the emperor with no clothes. Everybody knows this model is not that good, but because we're so feared that we don't have something better, we hold on to it, no matter how inefficient it is. And the patients are not happy, the doctors are not happy, the nursing staff is not happy. The only people that are happy in America are the insurance reimbursement people who are making a lot of money for inflating the cost of health insurance. Everybody else is pretty unhappy with the way the system is working. So this model, as you've heard from previous discussions, is a model that deals with personalization. It deals with preventive. It's predictive. It looks forward rather than back, predictive. And it actively involves participation with the person because it's their life. So they have to participate somehow in the outcome of their life. The 4 Ps, right? Dr. Leroy Hood, who is the developer of this concept called P4 medicine, he's a Nobel Prize winner in the States, links this in to what we're calling personalized lifestyle healthcare. And I have found that that term, personalized lifestyle healthcare, is an affiliative term that no matter how you translate it into whatever language, people can understand it. Because there's some sense, I call it a rule of reasonableness that people understand that lifestyle and environment has something to do with their health. They're not sure exactly how it relates, but they know that there's an interrelationship of some type. So here is a term that can affiliate all people from different walks of life, different ethnicities, both genders, different professions, all can subscribe to something common about personalized lifestyle healthcare. Now the question is, how much personalization is required? Because one could go to the extreme and say that this personalization needs to be every test that you could ever think of. And you suddenly have what we call the quantified human. If you go to Google the web right now and you put in the term quantified human, you will get all these hits of this new trend of people that want to measure everything in their body. Now with telemetry that we can measure our cardiac function and our respiratory gases and our blood sugar and our pulse rate with telemetry that's on our smartphones, everyone's saying, I'm going to measure everything and I'm going to become a quantified human. Well, that's probably unrealistic for most people in terms of both expense and time and effort. So when we talk about personalization, we're talking about first-level personalization. Rather than medicine being for the average, we're saying medicine is for the individual. Gee, that's a big concept, isn't it? Medicine is for the individual. When I think back to the way I was trained about 70-kilogram human beings. The average human being, how they would respond to a drug, how they would respond to a treatment, the average individual. And then you get into practice and you find you never find the average 70-kilogram human being. They're all uniquely different. And you say, well, hold on, this is a little bit of a, like I got false direction here. I was told I was going to be able to treat in the midst of the Gaussian curve, and now all my patients are outliers. So let me ask you, who protects the outliers? And I would say, at some focus or some level, every one of us in this room are an outlier on some principle, right? If you think of all your characteristics, some of us are outside the bell-shaped curve in the 2 standard deviations from the mean. We're outliers. Therefore, the medicine needs to be focused on the individual, not just on the average. It's a simple concept, But then how do you apply it? You have to do it stepwise. You don't jump to the conclusion of saying, well, let's just measure everything and hope for the best. You have to say, what are the big issues that we have to personalize on? And that's then starting that path or journey towards a different form of healthcare, a personalized participatory form of healthcare rather than a medicine for the average for acute illness-focused healthcare. Okay, so we have put together this institute in January of this year called the Personalized Lifestyle Medicine Institute. I would encourage you to go to our website. It's fairly robust. We have hundreds of blogs, lots of videos. We're trying to raise the level of understanding of what this means across different cultures. We've written an article that just appeared in a peer-reviewed medical journal on personalized lifestyle medicine. Here it is. And interestingly enough, when that was published and we did a National Library of Medicine search on that article, it came up that this is the first time in the medical literature, in peer-reviewed literature, that the term personalized lifestyle medicine has been used, which is very surprising to me. I would almost guarantee, however, this is not the only time this is going to be used. I think this is the front edge of a growing interest because this is where genomics meets lifestyle, meets environment, meets nutrition, meets stress management, meets exercise and activity, meets structure, meets bioenergetics. That's where the convergence is occurring with regard to all these disciplines that are now talking about what is the real origin of chronic illness. If it's not a single organism that is infective that causes heart disease or diabetes or arthritis or dementia, what are the variables that then cause this condition to be rising? And it's the intersection of all these factors together. So that's how we kind of vision in this article the nature of chronic illness from a personalized lifestyle healthcare perspective. There are all these layers of differing factors that interrelate. And around the bottom, you can say it's stress, diet, activity, and environment that interface or intersect. Now, a lot of doctors will say, theoretically, Jeff, I think this is an interesting model, but I really don't practice lifestyle medicine. I practice pharmacotherapy, or I'm a surgeon. So my question to those practitioners is, do your patients have a lifestyle? And of course, anyone that's honest will say, yes, of course they have a lifestyle. Then I would ask them, do you feel that their lifestyle has any influence in the outcome of the therapies you're going to administer? In other words, is it a variable that could influence the pharmacogenetics or pharmacokinetics of a drug or the repair rate after surgery, the recovery? And everyone would say, well, yes, I know it will. And then my response is, if those first two things are true and you purport to practice scientifically based medicine, wouldn't you want to hold that variable as fixed, meaning lifestyle to be controlled, so that the other things you're doing would be the independent variables? So you maximize the opportunity for a positive outcome with whatever you're doing. In other words, if you don't regulate lifestyle at all and counsel your patients, you are then taking the devil by the hindmost. You're basically taking the luck of the draw that whatever you do is going to be successful in that patient because you've just lost a big, important variable that you should be controlling. So how does this relate to epigenetics? Epigenetics, epi meaning above the genes, right? So if we think of the genes as the hardware, our genome is the hardware that codes for our being, then the epigenome is the software that tells how the hardware is going to run, right? You ever think of that analogy? So our hardware is our genome. The software that lays on top of it is our epigenome that then says how our genome is going to be expressed. Now let me give a little bit more to that. Within our book of life, which is called our genome, in humans there are 23 chapters. 23 chapters in that book of life. Each chapter, which is called our 23 pairs of chromosomes, is written half by our biological mother and half by our biological father. Within each of those chapters are hundreds, if not thousands, of stories, and those stories are called our genes. Those stories are written in 3-letter words with 4 letters in the alphabet, right? Within any cell of your body is the lexicon for any story of any cell at any age of your body. Therefore, if our book of life was being continuously read in its entirety all the time, we would be a mess, right? You all would agree with that. Because within your book of life, you've got the liver story, you've got the heart story, the kidney story, the brain story. Every tissue in your body, every organ is in there. So the liver only really wants to read the liver story, hopefully, not read the brain story. So what directs then the book of life to be read in different ways in different parts of your body? It's called epigenetics. Now, it turns out, and this is where the story gets very fascinating, that within the last 20 to 25 years, it's been discovered that, yes, in developmental biology, most of these marks that are put on the genes, these epigenetic marks, are immutable. They're fixed so that the liver cell will say the liver cell and not change over to be some other cell type. But there are some that are called metastable epialleles. Metastable epialleles. Now, there's still some discussion about how many of our 20 or 25,000 genes are epistable meta-alleles, but let's say just for the sake of discussion, it's somewhere around 100. These particular epistable meta-alleles can be put on and put off. They can be what I call marked or unmarked over the course of living. So our environment can modulate our genome messages so that there's some variability how it's expressed based upon the environment in which we live. Now you're going to say to me, that sounds very reminiscent of Lamarckianism. You remember Jean-Baptiste Lamarck? You're familiar with this great debate that he had with Charles Darwin? Probably. Here are our two figures, right, in history. So there was this battle about adaptation versus natural selection. Now, who won this battle? Clearly, we know Charles Darwin won the battle because every one of us in this room is a student somewhere in our educational history on Darwinian natural selection. This is how organisms evolve, is through this process. And adaptation, meaning you put an organism into an environment and it changes, was kind of washed out of the system as a thought because it was not seen to be as viable in understanding evolution of the species as was that of natural selection. But it's interesting how we go back to the future in science often. Back to the future. We find that we go back and we re-explore something and we say, oh my word, maybe we threw the baby out with the bathwater too quickly. Maybe there's something to this concept of Lamarckianism or adaptation at a fine-tuning level that we now call epigenetics. So this period of 1744 to 1849, this is the period Lamarck was born slightly before Darwin, but they fought out this philosophical battle only now to be kind of resurrected today in the 21st century. So you've seen these articles in previous speakers that are bringing up to the public view the concept of epigenetics. You are not just your genes. You're what your genes express. Let me say it another way. Our book of life, called our genome, is pluripotential. Pluripotential, meaning there are many stories in our book of life. Some stories are like a Greek tragedy. They're not so good. Other stories are like karmically blissful love stories. They're wonderful. The question is, what stories do people want to read out of their book of life? We call that genetic expression. And genetic expression is regulated by epigenomic marks. How the environment is influencing what part of our genome will be read. This is much more than related to the opportunity, what I would call plasticity, genetic plasticity. What I learned in school, which is a pathophysiologically focused medicine, is genetic determinism. Let's use an example. Angelina Jolie, celebrity. If I say Angelina Jolie, most of you have this kind of mental image probably that comes up immediately. The most recent might be of which her courageous decision to have what I call the most heroic preventive medical procedure that can be done, called a double mastectomy, because of her double recessive homozygous BRCA1 and 2 gene, which has then opened up a whole discussion among women around the world about, should I be tested for BRCA1 and 2? And if I had this, would I make the decision, given that its penetrance into the phenotype of breast cancer is over 85%, would I make the decision to follow her lead and have a double mastectomy preventively? Now let me say something about this. The woman who discovered the BRCA1 and 2 genes, a brilliant investigator back in the 70s at the University of California at Berkeley, what she found when she did her work was there was a high correlation between breast cancer and this homozygous recessive double allele of BRCA1 and 2. However, the penetrance, in other words, the genotype converting into the breast cancer at that point for women born in the 50s and 60s was 47%. That same genetic characteristic today, the penetrance into breast cancer is 87%. Now what changed? The genes did not change. The environment in which the genes are bathed changed, almost doubling the penetrance into the phenotype called breast cancer. Now, by the way, I'm not suggesting from that little model that I would counsel women one way or the other whether they should or shouldn't have a mastectomy based upon the information of the BRCA genotype. But what I'm saying is, even a high prevalence condition that we say, oh, that's a genetic condition, has a lot of variability based upon the environment in which the person finds themselves. Am I making sense here? Do you understand where I'm going? Okay, so as the Journal of the American Medical Association says, and this is actually authored by a colleague of mine, Andy Feinberg, he said, including epigenetics into epidemiological studies helps to explain the relationship between the genome and the environment and may provide clues for modifying effects in disease prevention and therapy, because it allows us to understand better how that person's genes could be expressed as disease. Or saying it another way, maybe a more provocative way, there is no such thing as a gene for diabetes, heart disease, dementia, Alzheimer's disease, autism. Choose your disease of the month. There is no such thing as a gene for any of those. In fact, I would even go so far, there may not even be such thing in the purest sense, a disease. A disease is a dysfunctional response of that person's genes to their environment. And atheroma is not just caused by a high-fat diet, it's caused by the person's genes in their arterial epithelium responding to the environment in such a way as to produce an untoward reaction of cellular proliferation that later becomes an atheroma that we call a pathology. And for convenience, we name it and blame it and call it coronary vascular disease. But in the purest sense of the word, diseases don't exist. What exists are dysfunctions. Now, if I'm not rattling your cage now, I never will. I cannot be more provocative than what I'm saying now, because this is fundamentally attacking the underpinning of the way we were all trained. Now, why am I doing that? Am I just a naysayer and I just want to go around kind of provoking people to anxiety? I'm doing it because only through inspection of our model, I think, could we break through to actually do something that would reduce the burden of chronic illness. Because if we keep moving away around the deck chairs on the Titanic, the ship still sinks. The band plays on. We have to reconsider the fundamental ways that we view illness in order to come up with a model that's going to remediate this rising tide of age-related chronic disease. You can't just, someone said, we have to change not only the medicine we do, but how we do medicine. So it's not just universal access to health care, it's what kind of health care? What are we going to offer for all these dollars or lira that we're putting into the system? Okay, so the mammalian epigenome is really kind of the signature of how our genes are going to express themselves in the environments in which we have been bathed, starting at the moment of fertilization of the ovum, maybe even preconceptually. Let me say it again. Maybe even preconceptually, because there are epigenomic alterations in sperm DNA and epigenetic modifications in maternal ovum DNA. So maybe even before the sperm met the egg, we're already setting the stage for certain types of processes of genetic expression later in life. So you saw this graphic before. To me, this is the most beautiful bit of jewelry of artistry in the whole world. Even Michelangelo can't beat this in terms of its beauty. And that's how this legacy we have in our most treasured book of life is protected against injury in what we call the nucleosome. It's not just DNA that's strung out and stuck together like a cord in a ball. It's actually wrapped up in this jewelry with in this nucleosome structure so tightly and so securely that you have to have very specific library cards to get to read one book at a time, right? It only unwraps itself under certain conditions to allow certain stories to be read in the book of life. Now that model of what can be read is related to the epigenetic marks put on the library, on the bookshelves. So the way this whole configuration occurs, the so-called compaction of our DNA, is not just there to protect it against injury, it's to protect it against being misread so that you don't deface that. That's called genomic instability when you start defacing and having low respect for your genes. What does that mean? Smoking, too much alcohol, drug addiction, not getting proper nutrition. That's called low respect for the most precious commodity we own, which is our genome. So this chromatin, this condensed nuclear material in the human, is bigger in size than any other plant or animal. So when the human genome study was being done to decipher the code of life in humans, it was assumed that we would have more genes in our book of life than any other plant or animal that had ever been studied because of the size of this material and the complexity of the human. So the presumption was that it was going to be found that we would have about 100,000 genes coding for our book of life within the human genome. And the reason they thought about 100,000 was the size of the chromatin material and the fact that there are plants like rice that have almost 60,000 genes. And we have to be more sophisticated than rice. By the way, the Pinot grape has 35,000 genes. So it was a big, unexpected surprise when it was announced in the Rose Garden upon the deciphering of the human genome that we had between 20 and 25,000 genes, half of that of the rice. How could that be? In fact, the homology of the human genome was 97.6% identical to the chimpanzee genome. That's too close for comfort for a lot of people. How could this be? It could be because the bulk of that material you're seeing there that's in the chromatin, the nuclear material, is not coding for protein. It's not genome. It's what was called for years junk DNA. Now, for 20 years, I've been giving seminars to doctors saying, this term junk DNA, we're going to come back later to think how stupid we were for calling that junk DNA. Because we have this tendency to think if we don't know exactly what it's doing, it must be not useful somehow. And so people called this relic DNA, like throwbacks from viral infections that got inserted somehow. We never got rid of it. It was trash. You know what the junk DNA is now? It's not junk at all. It's where the information processing centers for the reading of all the genome is found. It's the executive center. It's the promoter regions of genes. It's these things that say how genes are going to be read multiple at a time. And in fact, we have more multiple at a time readers, executive centers, than any other plant or animal. Think of how many permutations and combinations you could calculate of reading 20 to 25,000 genes multiple at a time. It's virtually an infinite number of arrays. So the diversity of humans for other animals or plants is the way we read our information, not just the information itself. Now, here's the key of this whole story. That information that's encoded in what used to be called junk DNA is the information that's being communicated with by the outside world through this regulatory process called intercellular signal transduction, or this kinase network of 540 different enzymes that take information from outside the cell through things like G protein coupled receptors and signal through the cytoplasm of the cell, through the nuclear envelope, through nuclear transcription factors, to induce certain portions of the genes to be turned on and read under certain environmental conditions. Now, if I'm not lighting up your fire in this discussion, then I'm not a very good presenter. Because this is a revolution. What I've just given you in this last 5 minutes, in very quick summary, work of tens of thousands of competent scientists and clinicians, is a paradigm-shifting concept that makes the biology of the last century like barber surgeons of the 19th century. It completely changes the landscape of how we view the origin of disease, the presence of health, and the diversity of function among people who have still 10 fingers, 10 toes, a nose, and 2 eyes. It completely changes the diversity of response. And it opens the door to make personalization more than just a term. It actually introduces it as a concept within medicine. So this access to genetic information is triggered through this promoter regions of genes, which are used to be in the junk DNA, that's regulated in part by our epigenomic marks that occur through, in part, environmental experiences. And that then leads to the regulation of gene transcription and ultimately even to the regulation of post-translational protein modification like phosphorylation, ubiquitination, or glycation, oxidation. All of those processes are regulated by this complex orchestration that's in part regulated through our epigenome. And what we know is that these genes that are turned on and turned off in their promoter regions, this what used to be called junk DNA component, can be modified to either allow the messages to be read or to be silenced. And one of the most important gene silencing things that we've learned is methylation of the promoter regions of genes. These GC repeat units that are at the front end of a gene that are like the on-off switch, when they're methylated, it says, can't read here. It's like putting a paper clip on your book, on some chapters of your book saying, sorry, you can't read this is X-rated. Or these promoter regions can undergo different effects like acetylation, which is like a sticky note on your book of life that says, oh, read here, this is a great chapter. So the sticky notes and paper clips, which are these epigenetic marks, determine how genes are going to be regulated to express their function. So let's say that we put paper clips, meaning don't read messages, on tumor suppressor genes. That probably wouldn't be so good, right? Or let's say that we put sticky notes, meaning read here, on oncogenes. That wouldn't be so good either. So it's the balance of events in our environment that put these messages on our genes that may cause cells to replicate, undergo dedifferentiation, metastatic events, cellular proliferative events that we associate with atheromas and cardiovascular disease, immune dysfunction that we call autoimmune disease. These are epigenetic manifestations of marks that were put on our genome saying, read here. Now let me use an example. If I asked you the question, what is the second most frequent diagnoses as a family of diseases for early stage disability and premature death, what would you tell me? You would say, well, let's see, we have heart disease as number 1, and I heard that cancer is number 2, but would you be surprised if I told you that cancer is actually not number 2? That emerging to be number 2, when you put them all together, 88 different diagnoses are called autoimmune disease. And autoimmune diseases are rapidly rising in prevalence in the developed world. Why? These are immunological disorders that some people said, these are genetic. Multiple sclerosis, systemic lupus erythematosus, myasthenia gravis, rheumatoid arthritis, Crohn's disease. These are genetic. Well, would you be surprised to learn that there is actually no strong penetrance of specific monogenetic characteristics of any of those diseases? There are certain immunological characteristics in the genes that give rise to certain susceptibilities, but families in which you find autoimmune disease, one family member might have MS, another might have rheumatoid arthritis, and another might have systemic lupus. They don't get the same disease. They have a prevalence of disturbances of their immune system that's hypervigilant to their environment. Then would you be surprised if I told you that many of these autoimmune diseases are promoted or aggravated by specific drugs and chemicals? Why is that? Because their immune system recognizes that as a foreigner, and could they be the yellow canaries whose immune system are specifically more sensitive to these chemicals, so they get these immunological imbalances before the general at large? But if our environment becomes damaged enough, might not we all have these immunological problems as a spreading effect? In other words, what's the tip of the iceberg? Once you start asking these questions, I'm telling you, it's like having a virus in your nervous system. You can't escape from it. It lives with you 24/7. This has been for me 30 years or more of constant living with this concept, saying the why. The why. What is going on? What's the nature of this interaction between the genome and its environment that is leading to the prevalence of certain new disease prevalences that were previously not seen, but are now penetrating into all sorts of age populations globally? Use autism as an example. There's a good example. Now we'd say, well, it's just better diagnosis. Certainly that plays a role. But when I was in medical school, we were told that autism was 1 in 10,000 births in America. There is a sign now on our freeway in Seattle, Washington, which is where I'm from, that says in the state of Washington, 1 in every 320 births is an autistic birth. Now that's 10,000, 1 in 10,000 to 1 in 300. That's more than just better diagnosis. There is something going on here in the environment that has created this unique constellation of what we call autistic spectrum disorders. These are the kind of questions we need to be asking, not just producing more autistic treatment centers. We need to be asking where these things come from. So this histone code I'm describing is just like our genetic code. It's laid on top. It's the software that is our acetylation, methylation, phosphorylation, ubiquitination epigenetic marks that regulate how our genes are read. And the point I want to make here is that these epigenetic marks are put there due to environmental exposures. The most obvious of which clearly is methylation. Because methylation, every methyl group that's transferred in our body comes from the same physiological process, which is called the folate cycle. 5-methyl tetrahydrofolate going to S-adenosylmethionine is the source of all methyl groups. So you might ask, well, what happens if you have animals and you put them on a folate deficient diet? Are they more susceptible to cancer? And yes, they are. They have 10-fold higher incidence of cancer if you take a folate deficient group of animals and expose them to a known low-level carcinogen than exposure to the carcinogen in the same animal species that's properly folate nourished. Why? Because if you're folate deficient, you're methyl deficient. If you're methyl deficient, you can't silence the promoter regions of your oncogenes. Are you seeing where I'm going here? This is the front edge of a whole new way of looking at the origin of metabolic disease, is through this connection of genes to environment. Okay, so we can silence genes by methylation, and we activate gene expression by acetylation, and these are tightly regulated processes that are interdependent with the environment in which the person finds themselves: nutritional environment, stress environment, toxic environment, lifestyle environment. All these things play roles and laid on top of the genes. So then if we were to apply this to the wonderful presentation that Gasparini shared with us about the gastrointestinal effect, I loved when he said that he believes the GI system is like the center of the universe because it's the interface between the outside environment and the inside environment. Because that's where a lot of epigenetics is occurring. It regulates then functions and signals that go out to the rest of the body. And the person that I really take my lead from that was probably the most important leader in this is Elie Metchnikov, who won the Nobel Prize in Medicine, as you probably know, in 1900 when he was the director of the Pasteur Institute. He took over for Louis Pasteur when he passed away. And he was credited as being the father of innate immunity. You know, the story goes, I don't know if this is true, I wasn't around then, but the story goes that he was on the southern coast of France, and he picked up a starfish, and he took a sea urchin quill, and he punctured the starfish, and then he looked with his magnifying glass, and he saw these, what looked like microbes, coming to the origin of attack, where he penetrated the starfish. And of course, this is the age of microbiology, so he's thinking, oh, these must be bacteria that are coming. And then when he examined, he found, no, it wasn't bacteria. These were white blood cells that were migrating to that point of attack, and that was the origin of the discovery of, he termed phagocytosis, innate immunity. So that work that Metagenics did led him ultimately to kind of connect together clinically immunity with bacteria, with the gut, and ultimately he wrote this book, which was finally translated into English in 1902. I have a first edition copy, actually, I'm very proud of, called Prolongation of Life. And in that book, he actually talks about the way that you promote health and treat disease is by instilling by enema Lactobacillus bulgaricus, a yogurt-cultured bacteria, into the rectum. Because he felt that gut health was directly related to physical health and aging. This was 1898 in French, 1902 in English. So this legacy goes quite a ways back, and I don't think I need to, because you've already had this wonderful discussion with a person who knows 10 times more about this than I do, so it'd be a little presumptuous of me to try to teach you gastroenterology when you just had gastroenterologists of the acclaim of the two of my former presenters here. So what I really just need you to recognize is that the GI tract is the seat of immunity. 70% of our antibodies are produced by the enteric immune system, the so-called gastrointestinal associated lymphoid tissue, or GALT. And we recognize that over 50% of the immune system mass is clustered around the gut. Now, people have asked me the question, why would you think that the gastrointestinal tract would be so connected to the immune system? Was it that the universal designer of the body just didn't have a better place to the immune system, so he or she just stuck that onto the intestinal tract? And the answer is no. Think of what happens in the course of living. An average person will consume between 10 and 15 tons of food in the course of their life. The majority of that food, unless they're cannibals, are foreign molecules. Right? They're foreign molecules. We don't eat ourselves, so we eat foreigners, plants or animals. Our body has to translate that foreign information into friendly information, both by digesting these big molecules to small things like amino acids or monosaccharides or monolipids, but it also will absorb some particles, some fragments like polypeptides. So it had better have a way of diffusing that information in those molecules before it gets into the blood, or the body's going to have foreign reactions. So that's the immune system that clusters our intestinal tract. So that's, we would call it very good. But it can also be our foe, because if you get an accelerated or aggravated response to your digesting contents, now that immune system can send a signal, how? Through the portal blood. Remember, you heard earlier that there is a direct connection between the GI illness and the liver. The hepatobiliary digestive system is one big system. Why? Because that information that comes from an aggravated gut immune system travels through the hepatoblood directly to its first site, which is the liver. The liver, 10% by weight, is made up of embedded white cells called Kupffer cells. So the information coming from the gut goes to the relay race player called the white cell, which then does what? It transfers it on by producing its own inflammatory messengers through macrophages and monocytes that travel in the blood. And where do they end up finally? They end up all over the body. Like they go through the blood-brain barrier and they affect the glia. The glia is the brain's immune system, the microglia. Or they go into the adipocyte mass, the fat mass. So you get macrophage infiltration of the adipocytes. And what are these macrophages going to say to the adipocytes, the fat cells? They're going to say, you know, I'm pretty kind of upset. Someone told me I ought to be aggravated. So I'm going to produce inflammatory cytokines, And you, adipocyte, then should upregulate your genes to produce what? Adipocytokines. And they have names like tumor necrosis factor alpha, interleukin 6, ghrelin, resistin. So they start talking back. Now the fat cell says to the rest of the body, what? I'm fed up and I'm not going to take it anymore. So let me ask you a question. Does obesity cause diabetes and heart disease and dementia? No. No, it does not. Last week, a study was published, 150,000 patient experiences in an epidemiological study showing there was no correlation between body mass index and chronic disease unless the individuals had poor fitness. If they were high BMI and they had poor cardiovascular or cardiorespiratory fitness, there was a very strong 5-fold increase in chronic illness. If they were high BMI, however, and they had high fitness, there was no statistical correlation with chronic illness. Let me give you another factoid. We're doing a lot of work with Roux-en-Y gastric bypass surgery. These are morbidly obese patients, BMIs of 40 to 45 to 50. Most of these individuals are polymedicated. They're taking cardiac arrhythmics. They're taking hypocholesterolemics. They're taking hypoglycemics. They're taking, let's see, what else? Anti-inflammatories. So they might be 6 or 8 different medications. They undergo this gastric bypass resective surgery. Within 7 to 10 days post-op, their BMIs are still 45 to 50, and most of them are completely off their medications. Their blood sugar is normalized. Their lipids have normalized. Their inflammatory markers have normalized. Now, if fat causes those problems, how do you rationalize this? Because there are 2 types of body fat. There is friendly fat with a big smile, and there is angry fat that's producing inflammatory mediators. Now, there is a correlation between elevated BMI and angry fat, but they're not one and the same. So when someone says to me, obesity causes diabetes, I say, prove it. Obesity in and of itself doesn't cause diabetes, but angry obesity definitely causes diabetes, or I would say it another way. I would say there's an underlying metabolic disturbance for which obesity and diabetes both can be the outcome. And this is one of the reasons I believe we've been so unsuccessful in treating obesity, because we're going after it wrong. We're saying cut out the calories. It's a simple thermodynamic model of the first law of thermodynamics, energy in versus energy out. Well, that's partially true, but what you forget about is the physiological machine in between called the human being. We're not a calorimeter. And that control of the physiology is what regulates the outcome called health or disease. It's not just the body mass in and of itself. It's the personality of the body mass in the way it's influencing physiology. So how do you then get a handle on that? You treat the functional disturbance, and lo and behold, the body mass index normalizes, as do the metabolic conditions normalize. Just like what happens with Roux-en-Y gastric bypass without the problems of malabsorption and all the other attendant risks that you get from surgery. We need a whole different strategy to manage the epidemic of obesity. It's not just putting people on diets. I would suggest there will never be a diet that will be successful to manage this epidemic. Okay, so maybe I've done enough here. Maybe I've provoked you enough. I'm known as the grand provocateur, so I can only probably do so much cage rattling before you've had enough, you feel uncomfortable in your chairs. But I believe what you can see that I'm trying to get you to understand is the digestive system is a great place to start. Yes, you might say, well, there's a million things we might want to know and personalize, but let's start first with the gut, because that's where a lot of the action is initiated. This is Back to the Future with Dr. Metagenics, and let's start with gut restoration. That's why, as Dr. Ungara was talking about, we developed this 4R program: remove, replace, reinoculate, and repair, because it's a simple way for a clinician to start dealing with the seat of imbalance that leads to dysfunction. And it cuts across all these diseases, from diabetes to dementia to fatty liver to arthritis to heart disease. So it's a place to start. It's not the be-all and end-all. It's not the end of the journey, but it's a good way to start the journey towards personalization. So if you start looking at both the local effects of the array of diseases that are associated with GI dysfunction, and then the systemic effects It's a pretty good laundry list of things that are related to the major threats to health in the aging individual. So I have a lot more slides, but I think this is probably a good place to allow you to ask some questions and have at me here. So thank you for your attention, and I'll take whatever questions.